Hello everyone. Let's find out the difference between photosystem 1 and photosystem 2. In short, we also use the term as PS1 for photosystem 1 and PS2 for photosystem 2. Now, these both topics are related to the topic of photosynthesis in plants. So as we know, in case of photosynthesis, plants make their own food. In that case, they need sunlight. And it depends that of which wavelength the sunlight is. On that basis photosystems are activated. So basically the photosystem 1 and photosystem 2, they excite their molecules or some other things on the basis of the sunlight they are getting, on the basis of its wavelength. Now let's have a look on some differences between photosystem 1 and photosystem 2. So firstly, remember that in case of photosystem 1 and photosystem 2, these two things are not based on their functions. Basically, when we talk about the photosynthesis, then in this case, the photosystem 2 is firstly active and later on photosystem 1 is activated. Their name is here the name photosystem 2 and photosystem 1 are named the according to their discovery basically. So in case of photosystem 2, in this one its location is in the thylakoid membrane of the chloroplast near the grana. So this one is located in the thylakoid membrane, while if we are talking about PS1, then it is also present in the thylakoid membrane, but typically it is also found in the stroma part of the thylakoid regions, where are less concentrated in the grana stacks. Now, in the second one, we are talking about PS second. It is primarily responsible for the initial steps in the light dependent reactions. So it helps in light reaction. It absorbs the light energy and then it uses it to split the water molecules, and it will split the H2O water molecules into oxygen and protons and electrons as well. So basically here photolysis of water occurs. While in case of PS1, it receives the electrons which are transported from the PS2 and then uses light energy to excite these electrons. The energy from the electrons is used to reduce NADP positive, which is reduced into the NADPH, which is then used in the Calvin cycle. Now let's have a look on the wavelength as well, which is most important part, that the PS2 absorbs light best at the wavelength of only 680 nanometer. That's why it is also known as P680. While in case of PS1, it absorbs light best at a wavelength of 700 nanometer. Hence, it is also known as P700. After this, after absorbing the light, the PS2 passes this excited electrons to the plastoquinone. In short, we use the term PQ for molecule, and basically this PQ is an electron acceptor which transfers them to the cytochrome B6F complex. And in case of PS1, PS receives electrons from the electron transport chain after the PS2 and passing them to ferredoxin, which is also electron acceptor, and which then help in the reduction of NADP positive to NADPH. In case of PS2, it is responsible for the oxygen evolution. Why? Because here we know photolysis of water is done. So basically when water molecules are broken down, then they will release hydrogen, or we can say protons, and oxygen as well. So here oxygen evolution will be there, while in case of the PS2, it does not directly participate in oxygen evolution. After this, if we are talking about the PS1, then it contributes to ATP synthesis indirectly only by creating a proton gradient across the thylakoid membrane. Later on, it drives which drives the ATP synthesis. So it is not directly related to the ATP synthesis. While if we talk about the PS1, so in this one, it contributes to the proton gradient also and help in ATP production, but primarily in the form of NADPH. After that, NADPH can be converted into the ATP. So basically from one NADPH we can produce about 3 ATPs. The last point here is that the electrons for PS2 come from the water molecule, while the electrons which have to be consumed in the PS1 come from the electron transport chain, which are originating from the PS2. So these were some basic and major differences between PS2 and PS1. Now let's have a look on the image so that it will be more clear to you. So here you can see firstly in this one, here photosystem second is written and after that photosystem 1. So first of all, photosystem second is activated when it get the sunlight of P680, or we can say 680 nanometer. After that it will transfer its H positive, or we can say protons or electrons. Sometimes may be possible, in some books it is written as proton, and sometimes it is written as electron, but here we are considering it about the proton. So here H positive is passed from plastoquinone to cytochrome B6F and then to PC, after that P700. So when the light is absorbed at this P700, that means at 700 nanometer, then it will get excited, and after that, after getting to all these parts, the process will be completed. So these are some basic differences between photosystem second and photosystem 1.